Everyone has experienced this, whether you want to admit it or not. Yes, diarrhea. In some parts of the world, this is no joke. In areas of poor sanitation, the bacteria Vibrio cholerae can be transferred from dirty water into the human intestine. Here, it causes profuse diarrhea, extreme dehydration, and in severe cases, death. The World Health Organization gives records of 100,000 to 120,000 deaths per year as a result of cholera. What we are interested in is how this tiny bacteria can survive in both marine environments and the human intestine, and importantly, how it transfers between the two. In order to do this, Vibrio cholerae secretes a protein known as GBPA. This three-dimensional protein has 4 binding domains. These domains allow it to bind to exoskeletons of marine organisms and to the surface of the human intestine. Domains 1 and 4 contain chitin binding regions, so in aquatic conditions they can bind to a marine organism's exoskeleton and mark it for colonisation. Domains 2 and 3 of the protein bind to the bacteria itself. In the human intestine, domain 1 of the GBPA protein binds to the mucus on the intestinal wall, marking it for colonisation. Domains 2 and 3 bind to the Vibrio cholerae bacteria.
We have been interested in chitin for a very long time. So chitin is actually the second most abundant biopolymer on the planet, next to cellulose in the plant cell wall. And chitin is found in the exoskeletons of insects and also of crustacea, so marine life like prawns and shrimps and crabs. We noticed in the genome of Vibrio cholerae a really interesting protein that appears to have 4 domains, and at the N terminus and at the C terminus there were 2 apparent chitin binding domains, one of which, the one at the N terminus, that we had just determined the crystal structure of a few years ago, of a homologous protein called CBP21. And so we were now interested in trying to figure out what this protein was doing, because at the same time a paper came out showing that this protein somehow interacts with gut epithelium and also interacts with chitin on marine organisms. So we were sort of interested in exploring the link between these 2 chitin binding domains and the organism interacting with 2 different kinds of surfaces. So one of the things that Vibrio cholerae does is, as you know, it's the causative agent of cholera, and it's a unique organism because it's able to reside in 2 very different environments. One is a marine environment where it is stuck to small marine organisms, and by that means it's able to spread over a large geographic area. And the other interaction is via the gut epithelium. So basically, once it is in the human, it's able to adhere to and colonize the human gut. So this seemed like a really interesting protein because it's kind of got, you know, both functions incorporated in it, right? It's a linear protein with 2 domains at the end that were predicted to be carbohydrate binding domains. So it seemed like interesting to investigate whether this may contribute to colonization of both the marine environment and the human gut. We were interested in trying to just trying to get the structure of that chitin binding domain and trying to get a complex with the sugar. But it turned out we managed to express the full protein, so we can see, how these two domains are acted via middle domains, and that then led to the hypothesis that this might be important for binding to carbohydrates in the gut epithelium and in chitinol marine organisms, which we then went ahead to test with biochemical assays in collaboration with a group in Calcutta. So what started as a sort of a simple structural project actually blossomed into something much more interesting. Yeah. So in this paper, there wasn't only the structure; there were a lot of biochemical experiments as well, and these experiments showed that indeed these two domains are able to interact with chitin and with gut epithelium cells. So having the structure in principle would allow us to design molecules that block interaction with these types of sugars. Unfortunately, though, in our paper we only present the structure of the protein in its naked form. It's not actually complex to any of the sugars, right?